Good morning, everybody. Welcome back to Daily Lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I am Dr. Bobby, and we are dealing with the subject of molecular biotechnology, and we are on the chapter of plant growth promoting bacteria. The subtopics in this chapter are growth promotion by free-living bacteria, decreasing plant stress, increasing phosphorus availability, biocontrol of pathogens, siderophores, antibiotics, enzymes, ice nucleation and antifreeze proteins, ethylene, root colonization, nitrogen fixation, nitrogenase, components of nitrogenase, genetic engineering of the nitrogenase, gene cluster, engineering improved nitrogen fixation. Hydrogenase, hydrogen metabolism, genetic engineering of hydrogenase genes, nodulation, competition among nodulating organisms, genetic engineering of nodulation genes, nodulation and ethylene, phytoremediation, engineering strains that facilitate growth, engineering degradative plasmids, engineering bacterial endophytes, metals in the environment, summary, and review questions. Under natural environmental conditions, successful plant growth and development and high crop yield depends on the genetic constitution of the crop species. The development and high crop yields depend on the genetic constitution of crop species, suitable weather conditions and soil components, including the availability of nutrients, the absence of growth inhibitory substances, such as salt, the presence of certain beneficial microorganisms, and the absence of pathogenic ones, called phytopathogens. From phyto, meaning plant, some beneficial indigenous soil bacteria and fungi act directly by providing a plant growth enhancing product, and others indirectly. The latter organisms inhibit the growth of the pathogenic soil microorganisms, thereby preventing them from hindering plant growth. The direct promotion of plant growth usually entails providing the plant with a compound that is synthesized by the bacterium, such as fixed nitrogen or a plant hormone, although also these bacteria can facilitate the uptake by the plant of certain nutrients from the environment. The indirect promotion of plant growth occurs when plant growth promoting bacteria lessen or prevent the deleterious effects of phytopathogenic organisms, either fungi or bacteria, that is, they act as biocontrol agents. This activity is called antibiosis, and it either depletes a scarce resource required by the pathogen or produces a compound that impedes the growth of the phytopathogenic organism. Direct stimulation of plant growth and development by plant growth promoting bacteria can occur in several different ways. The bacteria can 1. Fix atmospheric nitrogen to ammonia that is used by the plant. 2. Synthesize siderophores that solubilize and sequester iron from the soil and provide it to plant cells. 3. Synthesize phytohormones such as auxin, cytokinin, or gibberellin that enhance various stages of plant growth. 4. Solubilize minerals such as phosphorus that are used by the plant. And 5. Synthesize an enzyme that can modulate the level of the plant hormone ethylene. Any particular plant growth promoting bacteria may utilize one or more of these mechanisms. Much of these recent genetic research directed at creating microbial strains with augmented plant growth promoting activity has focused on a few areas of study. Engineering of better biocontrol strains of bacteria to decrease the damage to plants from a variety of pathogens. This work is aimed at replacing some of the chemical pesticides that have become environmental pollutants. The use of bacteria to lower ethylene levels in plants. These studies are directed toward preventing high levels of ethylene from accumulating in plants, and thereby decreasing the damage to the plant from a variety of environmental stresses, including drought, flooding, salt stress, and the presence of pathogens. The molecular basis of nitrogen fixation. This topic has been investigated thoroughly to determine whether it is possible to increase the level of microbial nitrogen fixation and consequently lessen the current dependency on chemical fertilizer for crop plants. Root nodule formation by symbiotic bacteria. This process has been studied with the aim of producing genetically engineered bacteria that can outcompete naturally occurring symbiotic bacteria. Microbial synthesis of iron sequestering compound siderophores. These reactions are being characterized in the hope that it might be possible to produce beneficial strains that prevent the growth of a phytopathogenic microorganism or microorganisms. Manipulation of plant growth promoting bacteria to facilitate bioremediation, the use of plants to remediate contaminated environments. Current research in this area mainly deals with the plant growth promoting bacteria rather than fungi. This is at least partly due to the fact that scientists have found it difficult or even impossible to grow many beneficial fungi in culture. So not only is it difficult to manipulate them in the laboratory, it is also extremely difficult to obtain large enough amounts of these organisms for inoculation of crops. In the past, bacterial fertilization had a dubious reputation. During the 1950s in the Soviet Union, more than 10 million hectares, about 39,000 square miles of farmland were treated with diazotrophic nitrogen-fixing bacterial mixtures that consisted primarily of Azotobacter, Cruococcum, and Bacillus megaterium. In these experiments, about 60% of the time, yields of various crops were increased by 10 to 20%. However, these field trials were poorly designed and not replicable, so many researchers were skeptical about the validity of the work and tended to discount the use of bacterial inoculants as fertilizing agents on a large scale. In recent years, considerable progress has been made toward understanding many of the mechanisms employed by plant growth promoting bacteria. Thus, there is a much greater likelihood than in the past that the results will be predictable and reproducible. Growth promotion by free-living bacteria. Plant growth promoting bacteria include a wide range of bacteria that are free-living or that form a symbiotic relationship with plants, such as Rhizobium and Frankia. While numerous plants free-living soil bacteria are considered to be plant growth promoting bacteria, not all bacterial, I'll come to this table later, strains of particular genus and species have identical metabolic capabilities. Thus, for example, some Pseudomonas butida strains actively promote plant growth, while others have no measurable effect on plants. The major applications of bacteria for improving plant growth include agriculture, horticulture, forestry, and environmental restoration, phytoremediation. In the past 20 years or so, based on a better understanding of the mechanisms employed by these bacteria, and following a large number of successful laboratory and field studies, an increasing number of plants growth promoting bacteria have been commercialized. The mechanism most commonly invoked to explain the various effects of plant growth promoting bacteria on plants is the production of phytohormones. Research in this area has focused on the role of a class of phytohormones called auxins. The most common and best characterized auxin is indole-3 acetic acid, IAA, which stimulates in plants both rapid responses, such as increases in cell elongation, and long-term effects, such as increases in cell division and differentiation. Since both plants and plant growth promoting bacteria can synthesize auxin, it is difficult for researchers to distinguish between plant responses that result from bacterial auxin synthesis and those that result from plant auxin synthesis. This uncertainty notwithstanding, there is a considerable evidence to suggest that many plant growth promoting bacteria facilitate plant growth by altering the hormonal balance within a plant. In the early 1990s, it was discovered that many plant growth promoting bacteria contain an enzyme that can modulate levels of the plant hormone ethylene. This enzyme 1-amino-cyclopropane 1-carboxylate ACC deaminase cleaves ACC, which is the immediate biosynthetic precursor of the ethylene in plants. As depicted in figure 15.2, the bacterium binds to seed coats of plant roots and then sequesters and cleaves ACC. As a result, the level of ethylene in the developing or stressed plant is lowered. In many plants, ethylene stimulates germination and breaks the dormancy of the seeds. If the level of the ethylene remains high after germination, a problem that is especially acute when plants are under stress and root elongation is inhibited. Thus, the ACC deaminase that is provided by a plant growth promoting bacterium prevents the inhibition of root elongation, and consequently the plant produces longer roots during early development, resulting in a healthier and larger plant. In addition, many plant growth promoting bacteria synthesize IAA. The IAA that is produced by the bacterium and taken up by the plant that is not used to promote plant cell elongation or proliferation stimulates the transcription of the enzyme ACC synthase in the plant. A greater amount of ACC synthase causes an increase in the level of ACC, eventually resulting in an increase in the ethylene concentration. When ACC deaminase activity is present, it prevents the buildup of ACC, even in the presence of high levels of IAA, so that the ethylene level does not become elevated to the point where plant growth is impaired. In general, nitrogen fixation by a free-living plant growth-promoting bacteria probably makes only a minor contribution to the growth of a plant. In fact, not all plant growth-promoting bacteria are disotrophic, and many of those that are disotrophic fix only limited amounts of nitrogen. A number of plants use bacterial iron sidra-4 complexes to obtain iron from the soil. Without this mechanism, plant growth in many soils would be severely limited, as iron is an essential plant nutrient. However, while bacterial sidra-4s undoubtedly contribute to the nutrition and hence to the growth of plants, in many instances this effect is small. There is some controversy regarding the mechanism that plant growth promoting bacteria use to facilitate the uptake of minerals such as phosphorus by a plant. On one hand, the increased mineral uptake in plants treated with plant growth promoting bacteria may reflect a better developed root system and an overall healthier plant. On the other hand, experiments with azospirillum have shown that this organism enhances mineral uptake by secreting organic acids that can solubilize and bind some minerals. This is a milestone note, a model for the lowering of plant ethylene concentrations by plant growth promoting bacteria, courtesy to the authors B.R. Glick and D.M. Penrose and J. Lee, J. Theo in Biology, and in the edition in 1998. On page 63 to 68, published in 1998. Thank you, sirs. Courtesy to all of them. In addition to the profound influence that ethylene has on normal plant growth and development, it is a stress hormone whose synthesis is increased when a plant is subjected to any one of a variety of environmental stresses. These stresses include mechanical trauma, pathogen infection, extremes of temperature, drought, flooding, salt, and the presence of environmental contaminants. Following periods of stress, the ethylene that is produced by the plant often exacerbates the effects of the stress. This can lead to plant senescence or death. Any chemical or biological treatment that lowers the amount of ethylene that is produced by a plant as a consequence of an environmental stress should therefore also increase some of the resulting damage to the plants. It has been known for many years that certain strains of bacteria can promote the growth of plants. That this article was published, at the time that this article was published, many of the mechanisms involved in the promotion of plant growth by bacteria had apparently been elucidated. However, there did not seem to be any one mechanism that could reliably and reproducibly promote the growth of a wide variety of plants under a range of different mechanisms. This was in spite of the fact that some strains of rhizobium, some biocontrol strains, and some azospirillum strains had been commercialized, albeit to a limited extent. In this article, Glick and co-workers developed a conceptual framework to explain a number of empirical observations that the laboratory had reported beginning in 1994. In its simplest terms, the model that they elaborated suggested that some plant growth promoting bacteria that were bound to plant tissues could act as a sink for some of the ACC that was produced by the plant in response to various types of stress. Since ACC is the immediate precursor of ethylene in all higher plants, the model predicted that lower ACC levels before it could be converted to ethylene would limit some of the deleterious effects of a particular stressor on a plant. The model was tested in a growth chamber, then in a greenhouse, and eventually in a field experiment. It was found that plant growth promoting bacteria that contained active ACC deaminase could significantly decrease the inhibition of growth and damage to plants following exposure to either high salt levels, the presence of metals, or organic contaminants, phytopathogens, flooding, or drought. Moreover, workers in many different laboratories around the world have found that this approach works well with a wide variety of plants, including canola, tomato, lettuce, soybean, mung bean, Indian mustard, various grasses, wheat, pea, corn, and cotton. Thus, by either selecting or engineering plant growth promoting bacteria to express ACC deaminase, the productivity of a range of crop plants can be improved dramatically. As a better understanding of the mechanism used by plant growth promoting bacteria emerges, it will become possible to genetically engineer improved organisms that can stimulate the growth of a wide range of plants in a variety of environments. Decreasing plant stress. In addition to its effect on seed germination and root elongation, ethylene mediates a wide range of plant responses and develops developmental steps. Ethylene is involved in tissue differentiation, formation of root and shoot primordia, lateral bud development, flowering, initiation, anthocyanin, synthesis, flower opening and senescence, fruit ripening and degreening, production of volatile organic compounds that are responsible for aroma formation in fruits, storage, production, hydrolysis, leaf and fruit abscission, and the response of plants to biotic and abiotic stress. In some processes, ethylene is stimulatory, while in others it is inhibitory. The term stress ethylene describes the increase in ethylene biosynthesis associated with biological and environmental stresses and pathogen attack. The increased level of ethylene formed in response to trauma inflicted by chemicals, temperature extremes, water stress, ultraviolet light, insect damage, disease, and mechanical wounding can be both the cause of some of the symptoms of stress. Example, onset of wilting and increased senescence, and the inducer responses that will enhance the survival of the plant under adverse conditions. Often, a small amount or a small burst of ethylene is synthesized by plants within a few hours after environmental stress. This low level of ethylene acts as a trigger to initiate the biosynthesis of a number of plant defense proteins. Subsequently, some 2 to 4 days after the onset of the stress, the plant produces a much larger burst of ethylene. It is in this second peak of ethylene synthesis that is responsible for attenuating the deleterious effects of the stress. While chemicals have been successfully used to control ethylene levels in plants, many of them are either expensive or potentially harmful to the environment. Consequently, ACC-DM-minase containing plant growth promoting bacteria have been tested to determine whether they could be used as an environmentally safe method for lowering plant ethylene levels. Flooding is a common abiotic stress that affects many plants, often several times during the same growing season. Plant roots suffer a lack of oxygen as a consequence of flooding. This in turn causes deleterious effects such as wilting, epinasty, which is also called as epinasty, inhibition of leaf chlorophyll synthesis, which is called chlorosis, cell death, necrosis, and reduced fruit yield. Many plants respond to flooding by activating the transcription in root cells, as some of the genes that code for AIIMS of ACC synthase, the enzyme that converts the compound S-adenosylmethionine into ACC. This eventually results in an increase in the amount of ACC inside plant roots. However, since ACC oxidase cannot catalyze ethylene synthesis in the absence of oxygen, ACC is transported from the anaerobic environment of flooded roots to the aerobic shoots where it is converted to ethylene. The ethylene in the shoots cause plants to wilt, to lose biomass, and eventually, if the ethylene remains elevated for a prolonged time, to sneeze and die. Treatment of tomato plants with ACC-DM-minase containing plant growth promoting bacteria significantly decreases the damage suffered by these plants due to stress ethylene brought on as a consequence of flooding. These ACC-DM-minase containing plant growth promoting bacteria can act as a sink for ACC, lowering the level of ethylene that can be formed in the shoots and thereby protecting the tomato plants from a promotion of the damage caused by flooding. In addition to protecting plants from flooding damage, ACC-DM-minase containing plant growth promoting bacteria can also significantly decrease the damage to plants that is caused by drought, temperature extremes, high concentrations of salt, and a variety of environmental contaminants. For example, in greenhouse experiments, tomato plants treated with Ichromobacter pyochordii ARB8, which contains ACC-DM-minase, were able to grow better in the presence of 86 salt, which is usually inhibitory to plant growth, than with tomato plants grown without the added bacterium in either the presence or the absence of salt. This bacterium, which is isolated from a soil sample from the Arava region of the Negev desert in Israel, significantly lowered the level of stress ethylene produced by tomato plants in the presence of salt. More recently, several groups have shown that this approach can facilitate the growth of a range of crops in saline soils in the field, a problem that is endemic to about 25% of the world's arable land. Increasing phosphorus availability. While a number of plant growth promoting factors promoting bacteria can synthesize and secrete organic acids that can dissolve inorganic phosphate in the environment, these organisms can rarely break down phytate, the complex compound inositol hexaphosphate that is for the major chemical form of phosphorus within cereal grains and oilseed. Several plants can produce phytases, enzymes that degrade phytate. However, the activity of these enzymes in plant roots is generally low, so these plants cannot efficiently utilize the phytate that is found in the soil. A gene encoding the enzyme phytase was isolated from the fungus Aspergillus fumigatus. This gene was inserted using a transposon into the chromosomal DNA of a strain of the bacterium Bacillus mucilaginosis, which can dissolve phosphorus from calcium phosphate. The transformed bacterial strain can express and secrete active phytase. In greenhouse experiments, this transformed bacterium was found to be superior to the wild-type strain in providing phosphorus to tobacco plants cultivated in its presence. Importantly, the stimulation of plant growth that was observed in greenhouse experiments was also evident in the field, where the yield of tobacco plants increased by 19%. Notwithstanding these results, it may be some time before this work is commercialized because of political concerns about the use of genetically engineered bacteria in the environment. Biocontrol of pathogens. Phytopathogens are an ongoing and serious agricultural problem that can reduce crop yields by 25 to 100%. This is an enormous loss of productivity. Currently, phytopathogen damage to crops is generally dealt with by the use of chemical agents, although other treatments have also been employed for most bacterial diseases. Plants may be symptomless for prolonged periods before changes in environmental conditions, which favor the proliferation of the bacterial cause a rapid outbreak of disease. Under these conditions, severe damage can occur and destroy an entire crop. These field epidemics are difficult and costly to control. Many of the chemicals that are used to control phytopathogens are hazardous to animals and humans, and they persist and accumulate in natural ecosystems. It is therefore desirable to replace these chemical agents with biological control agents that are more friendly to the environment. One approach for the control of the phytopathogens is the development of transgenic plants that are resistant to one or more of them. See the chapter 18. Alternatively, some plant growth promoting growth promoting bacteria can act as a biocontrol agent to suppress or prevent phytopathogen damage. And a number of these biocontrol bacteria have been commercialized, as shown in Table 15.3. Plant growth promoting bacteria can produce a variety of substances that limit damages to plants by phytopathogens. They include siderophores, antibiotics, other small molecules, and a variety of enzymes. This approach is still at an early stage of development but appears to have considerable potential. However, the ultimate utility of a strategy based on a particular mechanism can be assessed only under field conditions. Siderophores. Iron is one of the most abundant minerals on Earth and is an essential requirement for living organisms. However, iron in the soil is unavailable for direct assimilation by microorganisms because ferric iron, or Fe3, which is the predominant form in nature, is only sparingly soluble. That is, its solubility is about 10 raised to minus 18 m at pH 17.4. This amount of soluble iron is much too small to support microbial growth. Consequently, to survive in this environment, soil microorganisms synthesize and secrete low molecular mass, 400 to 1000 Dalton, iron-binding molecules known as siderophores. Siderophores bind Fe3 with a very high affinity dissociation constant, Kd, so 10 raised to minus 20 to 10 raised to minus 50 m, and transported back to cell. Surface receptors, where it is taken into the cell. Once inside a cell, the iron is released and is then available to support microbial growth. Plant growth promoting bacteria can prevent the proliferation of fungal phytopathogens by producing siderophores that bind most of the Fe3 in the area around the plant root, the rhizosphere. The resulting lack of iron prevents fungal pathogens from proliferating in the immediate vicinity. Fungal phytopathogens also synthesize siderophores produced by plant growth promoting bacteria. In effect, the plant growth promoting bacteria outcompete fungal phytopathogens for available iron. Unlike microbial phytopathogens, plants are not generally harmed by the localized depletion of iron in the soil caused by plant growth promoting bacteria. Most plants can grow at much lower iron concentrations than microorganisms. In addition, some studies have shown that iron has been sequestrated. Bacterial siderophores is taken up from the soil by the plant to its benefit. Because the sequestering of iron by a siderophore producing bacterium can simultaneously prevent the proliferation of a number of different phytopathogenic microorganisms, sitrophore genes are being examined to determine whether they can be used to create more effective biocontrol inoculants. Sitrophores generally have three functional or ion-binding groups connected by a flexible backbone, often a peptide. Each function group usually presents two atoms of oxygen or less, commonly the oxygen atoms, the nitrogen atoms that bind iron. In chemical terms, the function groups are bidentate. Trivalent ferric iron can accommodate three of these groups to form a 6-coordinate complex. With some exceptions, the functional groups in microbial sitrophores are either hydroxymates or catecholates. Different combinations of functional groups may be present on a single sitrophore. Other functional groups include carboxylic moieties such as citrate and the ethylene diamine. In general, hydroxymate type sitrophores are typical of fungi, and catecholates, which bind iron more tightly than hydroxymates, are common in bacterial sitrophores. Plant sitrophores, on the other hand, are linear hydroxy and a minor substitute of iminocarboxylic acids such as mucinic acid and havanic acid. One bacterial sitrophore, called pseudobactin, has been estimated to bind to Fa3 with an affinity constant of approximately 10 raised to 25 liters mole raised to minus 4. All fluorescent pseudomonads, so named because they produce a sitrophore that fluoresces when excited by ultraviolet light, synthesize structurally related sitrophores that differ mainly in the number and configuration of amino acids in the peptide chain that makes up the backbone. The synthesis and regulation of pseudobactin in the plant growth promoting bacterium Piputida WCS358 has been examined in detail. Mutagenesis was used to generate a set of 28 mutants that were defective for sitrophore production. The two criteria were used for identifying the sitrophore deficient mutants. One was a lack of fluorescence under UV light, and two, inability to grow in the presence of bipyridine, a molecule that sequesters most of the iron in the growth medium. When most of the iron is unavailable, only a cell that produces sitrophores can grow. A clone bank of Piputida WCS358 DNA was constructed in the broad host range vector PL-AFRI and was introduced by conjugation into each of the sitrophore mutants. All of the resultant transformants were tested by complementation for restoration of fluorescence and or the ability to grow in the presence of bipyridine. 13 separately complementing cosmic clones with an average insert size of 26 kilobase pairs kb were identified. After detailed analysis, these clones were found to represent at least 5 separate gene clusters. One of these clusters has been studied further. It has a length of 33.5 kilobase pairs and contains 5 transcriptional units with at least 7 separate genes, thus like nitrogen fixation and nodulation in sitrophore biosynthesis. Is a complex process since each sitrophore is encoded by a number of different genes. Genetically engineered bacteria to produce modified sitrophores is not a simple matter. However, there may be other ways to improve the effectiveness of plant growth, promoting bacteria as biocontrol agents. For example, it may be possible to extend the range of iron sitrophore complexes that one bacterial strain can utilize so that a genetically altered plant growth promoting biocontrol bacterial strain could take up and use sitrophore synthesized by other soil microorganisms, thereby giving it competitive advantage. This was done by cloning the genes for iron CYP4 receptors from the one plant growth promoting control bacterium and introducing them into other strains. Antibiotics, one of the most effective mechanisms by which plant growth promoting bacterium can prevent phytopathogen proliferation, is the synthesis of antibiotics. For example, the antibiotic synthesized by Biocontrol pseudomonads include agrocin 84, agrocin 434, 2,4-diacetyl-fluoroglucanol, herbicolin, oomycin, phenazines, pyrrolatorin, and pyrrolonitrin. The biocontrol activity of a plant growth-promoting bacterium may be improved by providing it with genes that encode the biosynthesis of antibiotics that are normally produced by other bacteria. In this way, the range of phytopathogens that a single biocontrol bacterium can suppress can be extended moreover by limiting the growth of other soil microorganisms. Antibiotics secreting plant growth promoting bacteria should facilitate their own proliferation, since they will have fewer competitors for limited nutritional resources. In addition, genetic manipulation can be used to increase the amount of antibiotic that a bacterium synthesizes. The production of a number of antifungal metabolites that are produced by Pseudomonas appears to be controlled by a protein that acts as a global transcriptional regulator. Therefore, it should be possible to enhance antibiotic production by modifying this global regulation. For example, antibiotic production was enhanced after Pseudomonas fluorescens. Chow was transformed with a vector carrying the gene encoding the housekeeping RNA polymerase sigma 70. The modified strain was more effective at a protein but protecting cucumber plants against root disease caused by fungus Pythium ultimum. A single copy of the operon carrying all 7 of the genes encoded that encode the biosynthesis of antibiotic phenazine 1-carboxylic acid, that is PHZ-ABD-CD-EFG, was inserted into the chromosomal DNA of the plant growth promoting bacterial strain of P. fluorescens. The wild-type version of this bacterium, which does not synthesize phenazine 1-carboxylic acid, acts as a biocontrol agent against some fungal diseases, as indicated by a much larger zone of clearance of the fungal pathogen. And P. ultimum on solid medium. The engineered bacterium has a higher level of biocontrol activity than the wild type. Also, the phenazine 1-carboxylic acid producing bacterium prevented P. ultimum caused damping of disease in pea plants and soil. This work demonstrates the efficacy of this approach under greenhouse conditions. However, it remains to be demonstrated whether this altered bacterium is effective in the field. At present, there is still only one commercial available genetically engineered biocontrol bacterial strain. A modified version of Agrobacterium radiobacter K84 has been marked, first in Australia in 1989, and more recently all over the world as a means of controlling Crown gall disease, which is caused by the bacterium. Agrobacterium tumefaciens. This disease affects almond trees and stone fruit trees, such as peach trees. The antibiotic agrocin 84, which is produced by A. radiobacter, is toxic to A. tumefaciens. However, agrocin 84 resistant strains of A. tumefaciens can develop plasmid carrying if the plasmid carrying the genes for the biosynthesis of agro 84 is accidentally transferred from A. radiobacter. To avoid this possibility, the region of DNA responsible for transfer was removed from agro 84 plasmid PAGK84. As a result of this deletion, the A radiobacter strain retains the capacity to act as a biocontrol agent, but it can no longer transfer the plasmid to pathogenic agrobacteria. Enzymes. Some plant growth-promoting bacteria produce enzymes such as chitinase, beta-1,3-glucanase, protease, and lipase that can degrade fungal cell walls and cause the fungal cells to lyse. In one study, the incidence of plant diseases caused by the phytopathogenic fungi Rhizotonia solani, Sclerotium rolfesi, and P. ultimum was reduced by using beta-1,3-glucanase producing strain of Burkholderia cepacia. In another study, the antifungal activities of 3 different strains of the plant growth promoting bacterium Enterobacter agglomerans were attributed to a complex of 4 separate polypeptides that act together to degrade the chitin in fungal cell walls. When tested, these bacteria significantly decreased damage to cotton plants following infection with R. solani. Moreover, TN5 mutants of E. agglomerans that were deficient in chitinase activity were unable to protect plants against damage caused by a fungal pathogen, indicating that the chitinase was the active element. Many of the bacterial enzymes that can lyse fungal cells, including chitinases and beta-glucanases, are encoded by a single gene. It should therefore be straightforward to isolate these genes and transfer them to plant growth-promoting bacteria to construct strains that produce fungus-degrading enzymes. In one series of experiments, a chitinase gene was isolated from the bacterium Serratia marcescens and then transferred into Trichoderma hertzianum and Rhizobium meliloti cells. In both cases, the transformed microorganisms produced chitinase and displayed increased fungal activity. On the S. marcescens, chitinase gene was introduced into a strain of P. fluorescens that directly promotes plant growth. The transformant also stably expressed and secreted active chitinase, and effectively controlled the phytopathogen R. solani. Ice nucleation and antifreeze proteins. One of the ways in which some pathogenic leaf bacteria such as Pseudomonas syringae damage plants is by synthesizing ice nucleation proteins. These proteins, which are produced at low temperatures, are present on the surface of the bacterium and act as sites that facilitate the formation of ice crystals at freezing temperatures. As the ice crystals grow, they can pierce the plant cells and cause irreparable damage. The bacteria benefit from this damage by gaining direct access to the nutrients from the lysed plant cells. In the absence of ice nucleation, plant proteins on the leaf surface with overnight frost would not damage the plant because the water in a plant cell must usually be several degrees below the freezing point before ice crystals begin to form. That is, it must be super cold. One way to prevent freezing damage caused by P. syringae to susceptible crops such as strawberries is by spraying the plants prior to the frost with the mutant form of One way to prevent freezing damage caused by P. syringae to susceptible crops such as strawberries is by spraying the plant prior to the frost with a mutant form of ice-nucleating bacterium. Such a mutant, which may be constructed by either recombinant DNA manipulation or conventional mutagenesis and selection, lacks the ability to produce the ice-nucleation protein, and therefore ice crystals are not formed on the leaf surface. If a sufficient number of these ice-minus mutant bacteria are sprayed onto a susceptible plant, the mutant will displace the wild-type ice plus bacteria, thereby preventing ice nucleation. An important facet of the effectiveness of a biocontrol plant growth promoting bacterium is its ability to persist and proliferate in the natural environment. In areas such as Canada, Scandinavia, Russia, and the northern United States, these organisms must survive long cold winters and then grow at cool soil temperatures. In the spring, about 5 to 10 degrees centigrade, while microorganisms have a variety of adaptive strategies for thawing under adverse conditions. It may be possible to engineer organisms that are better able to deal with cold temperatures. Some soil bacteria that are also able to promote plant growth can both grow at 5 degrees centigrade and secrete antifreeze proteins into the surrounding medium and growth at low temperatures. A bacterial antifreeze protein regulates the formation of ice crystals outside the bacterium and thereby provides protection for the bacterium and the soil to the presence of bacterial antifreeze protein. Ice crystals still form, but their size is limited. In the absence of antifreeze proteins, ice crystals can grow to a large size and eventually puncture the bacterial cell wall and membrane, causing cell lysis. Ice crystals do not form inside the bacterium to any great extent. This is because at low temperatures, bacteria decrease their volume by pumping some of the water in from inside to the outside of the cell. Recently, the gene for a bacterial antifreeze protein was isolated and characterized. The strategy that was used to isolate this gene included purifying the protein, digesting it into small peptides with the proteolytic enzyme trypsin, determining the amino acid sequences of several of these peptides, and using those amino acid sequences to design polymerase chain reaction PCR primers for a portion of the antifreeze. We come to the milestone in a jiffy. Protein gene, following the sequence determination of the PCR amplified DNA, inverse PCR 15 to 60.16 was used. That's figure 15.16. So this inverse was used to obtain the remaining portion of the gene. It should be possible to transfer this gene to various strains of plant growth promoting bacteria to create strains that can persist and proliferate at cold temperatures. Although there is currently no evidence to link antifreeze activity with the mechanism that bacteria use to function at low temperature, it will be interesting to examine experimentally whether antifreeze protein activity is part of the adaptive strategy used by some bacteria for cold as well as freezing tolerance. Ethylene. Fungal pathogens not only directly inhibit plant growth, they also cause the plant to synthesize stress ethylene, which causes some of the damage sustained by plants infected with fungal pathogens. For example, it was well known that exogenous ethylene often increases the severity of a fungal infection, whereas ethylene synthesis inhibits significantly decrease the severity of a fungal infection. As mentioned earlier, stress ethylene that is synthesized in response to fungal pathogen infection is produced in two peaks. A small one that occurs within a few hours after fungal infection, and a much larger peak that occurs several days after fungal infection. The first peak that occurs several days after fungal infection The first peak turns on the transcription of genes that encode proteins that protect plants against the pathogen, while the second peak is deleterious to the plant. Ideally, it would appear to be advantageous to allow the plant to synthesize the first but not the second peak of ethylene in response to a fungal pathogen. This is readily achieved using ACC deaminase containing plant growth promoting bacteria, since high-level expression of ACC deaminase does not occur until several hours after the appearance of increased amounts of ACC. Thus, these bacteria do not alter the first ethylene peak but significantly decrease the magnitude of the second peak. A biocontrol bacterium strain, P. fluorescens Xiao, was transformed with the P-butida UW4-ACC-DR- gene, and the effect of this manipulation on the damage to cucumbers caused by P. ultimum was assessed. The ACC-DR- containing biocontrol bacterial strain that did not possess the enzyme. Not only did plants inoculated with the ACC-DR- transformed strains have greater root and shoot biomass than those treated with wild-type biocontrol strain, but also the number of seeds that germinated in pathogen-containing soil was larger. In addition, the ACC-DR- transformed biocontrol strain reduced the extent of soft rot or potato slices caused by the bacterial pathogen Ovinia keratovora subspecies keratovora in sealed plastic bags by 50% compared with wild-type biocontrol strain. In effect, ACC-DR- acts synergistically with other mechanisms of biocontrol, such as production of antibiotics or antifungal enzymes to prevent phytopathogens from damaging plants. Root colonization. Depending upon the mechanism that a particular biocontrol bacterium uses to thwart the damage to plants caused by pathogenic microorganisms, it can be advantageous for the biocontrol strain to bind as tightly as possible to the plant root. One way to improve root colonization by biocontrol bacteria entails overexpressing the bacterial triplet gene. This gene is normally thought to play a role in DNA rearrangements that regulate the transcription. A gene or genes involved in the biosynthesis of cell surface components. When the triplets gene was introduced on a plasmid into two strains of P. fluorescens, one that was normally a poor root colonizer and the other a good root colonizer, colonization of tomato roots was increased by approximately 28 and 12-fold, respectively. This work is an important first step in engineering more effective strains of biocontrol bacteria. Nitrogen fixation. Nitrogen gas N2 has, which makes up approximately 80% by volume of the air that we breathe, cannot be used directly by plants or animals to synthesize essential nitrogen-containing biomolecules, such as amino acids and nucleotides. Rather, it must first be fixed into ammonia. This conversion requires a high input of energy because the triple bond of N2, that is N is extremely stable. The energy for the biological fixation of nitrogen comes from the hydrolysis of large amounts of ATP. Similarly, the chemical industrial conversion of N2 to ammonia uses a considerable amount of energy in the form of high temperature and pressure. More than 100 million tons of fixed nitrogen is needed annually to sustain global food production. Synthetic chemically produced fertilizers account for about half of this nitrogen supply, and most of the remainder is derived from the diazotrophic bacteria. No eukaryote is known to fix nitrogen. Chemical fertilizers have helped considerably in increasing crop yields, but their continual use has led to pollution problems as a result of runoff to depletion of the nutrient reserves in the soil. Moreover, their cost has been rising steadily. These factors have been provided as an incentive for developing alternative sources of fixed nitrogen, including development of diazotrophic microorganisms such as bacterial filters. This lecture should be over in a minute and a half from now, and we should continue in the next lecture. A wide range of bacteria can fix nitrogen, and a number of them have potential as crop fertilizers. However, until a bacterial fertilizer has been shown conclusively to be as effective as a chemical formulation, there will be reluctance to change current practice, especially in those countries where the cost of chemical fertilizer is not significant relative to the value of the crop. For example, soybeans, which constitute the second largest crop in the United States in terms of both cash value and total acres planted, form a beneficial symbiotic relationship with the bacterium Bradyrhizobium japonicum. In this symbiosis, the bacteria that provide the plant with the fixed nitrogen and in turn receive photosynthetically fixed carbon from the plant. When plants inoculated with specific strains of B. japonicum, the final yield material can be increased by 25 to 50%, and the inoculated plants no longer require the addition of chemically fixed nitrogen. Now, most of these farmers who use this continue to depend on the naturally occurring strains in B. japonicum in the soil and chemical fertilizers. So that's it for this lecture here. I'll meet you in the next lecture shortly. Thank you. Good day. The most important